Namaste and a very warm welcome to India Science. I'm Nidhi Kumar. And as usual, we bring you today some vaccination news and our new program, Fact 5, where we know that there is a lot of good news on the vaccine front. And we also know that 0.002% have been hospitalized for adverse effects. Today we have with us as our expert Dr. Anil Murthu, who's the Director, Professor of Medicine at the Lady Harding Medical College and Hospital. Many thanks for joining us, sir. Namaste and welcome to India Science. Sir, सबसे पहला सवाल I'd like to ask you the first question is that, there's so many vaccine platforms that are under development. If we talk about the GOI, could you explain the science behind them as two more are coming out, which is Zydus and the Johnson one? Sir.
Have a whole spectrum of vaccine platforms using which various vaccines are under various stages of development all over the world, including our own country, as we know. Of them, the ones that are of relevance direct are, of course, the RNA platforms, the DNA platforms, which are using the RNA. You do not deliver the virus or any virus particle. You just develop the information, the genetic information carried in the RNA through various ways into the cell to code for that spike protein, which then expressed on the cell attracts the immune reaction, and that is restored into the memory and for the future use should the real virus come across. So these are the RNA and DNA platforms. Both are fully developed now. In our country, the ZyCoV-D is a DNA plasmid-based DNA vaccine platform, which is currently undergoing trials, phase 1 and 2 in Ahmedabad. Then the ones of direct relevance to us are two. The killed virus platform, which also goes by the name Covaxin, which is now in use under restricted conditions of clinical trial mode. Here, this uses basically a whole virus which has been grown in the labs using Vero cell lines. The virus is grown and killed, destroyed, so as to render it non-infective. It cannot cause infections when it comes into our system as vaccine. Rendered non-infective, dead. However, it has still the basic structure with all the four antigens, E, M, N, and of interest to us, the spike protein, S. All these four antigens around the envelope in a killed virus enter our system as the vaccine, and that provokes our immune system to respond and save it in our immune memory for a later day should the real virus attack us. This is the Covaxin, which has been released under restricted use. The third platform available today in actual terms, practical terms, is the adenovirus vector platform. This uses, it uses a DNA, which just that fragment of the DNA of the coronavirus, which encodes for the spike protein, the protein of interest to protect us. We take this and engineer it into another virus, which will be used as a carrier to take this fragment into our cell. And this carrier is an adenovirus, not a human adenovirus. It's a chimpanzee adenovirus, which is inactivated so as not to cause any infection in us. Anyway, since it's a chimpanzee virus, it does not really cause any infection in humans. There's a species barrier. So using this chimpanzee adenovirus as a Trojan horse, as a vehicle, this fragment of message which encodes for the spike protein is delivered into our cells through this vaccine. Once it enters, it provokes, it replicates, and produces only the spike proteins which are expressed on the cells. Antibodies develop, are stored, that memory gets stored in the T and B cells for the future use should the virus come across us again in the real conditions. So these are the main, and then there are component vaccines under development, the Novavax and all, which use just small subcomponents of artificially engineered virus fragments to induce the same reactions as us in us. So these are broadly the main platforms of relevance to our country today, and they are in use.
So between Covaxin and Covishield, the question is that even though it is being given to health workers primarily in the beginning, the Delhi figures today read at 54% only, and Tamil Nadu was at 22%. So why this vaccine hiccup? Why this vaccine hesitancy is my question.
See, what is hesitancy? We all have hesitancy to various things, and when do we develop hesitancy in real life, in any situation? When anything is new, the suddenness with which a new situation is thrust upon us under conditions of uncertainty and lot of fear and panic. What is the normal emotional reaction of any human being to any situation? It
Mm,
is
mm,
conservative. It is hesitancy. Let me wait and watch. Let me see what happens to him or her. Let me stay away. Let me look. Why me? Let the others go. Those are normal ideas and concerns and emotions that we
mm,
undergo. So hesitancy is to be expected. It's natural. It's human. And this relates
mm,
to our belief systems. What do we believe? How healthy we are, how vulnerable we are, how likely we are to get the virus or the serious impact should we get it, vis-a-vis the risks and benefits of taking a vaccine. Now, this is the initial, we are just the initial phase of the vaccination, and this psychological inertia is a given, it's a human given. It will mobilize itself. We will be able to mobilize ourselves out of this inertia as time goes by, and as we observe that things are panning out normal as they are. And to date, not a single death that has been reported is
mm.
on account of this vaccine. It has not been caused by this vaccine. Those are coincidental issues. As you just rightly said, of all the serious adverse events reported, a mere 0.004,
Mm.
0.004% have been serious enough to require admission in a hospital, and they too, all of them have been discharged. If you look at the totality of serious side effects, mild, moderate, severe, all, it is just 0.14%. 0.14%. Now, what do we focus on? This 0.004% of hospitalizations recovered now, or do we focus on the 99.999% protection benefit that is expected? I would say that's a matter of
Bilkul.
framing the perspective. So over time, this perspective will shift from that 0.004 to that
Mm.
99.999. That will happen. Initial inertia is expected. I think we will overcome it over time.
Absolutely, sir. So as we go from virus to vaccine, my next question is, who is an immunocompromised person? Can you list out that criteria, please?
Immunosuppressed and immunocompromised. Uh,
Right.
Slight difference, although in the same frame. Immunosuppressed are those whose immune systems have been weakened, muted, blunted on account of some extrinsic drug,
Mm,
steroids,
mm.
long term steroid intake,
Correct, correct,
anti
correct.
cancer drugs, drugs used for treatment of connective
Mm,
tissue disorders like rheumatoid arthritis and all, drug induced. Immunocompromised are usually these refer to inherent condition states like those resulting from, let's say, HIV, those who have people living with HIV, people who have cancers spread. So these are inherent internal states that create blunted immune systems. And then there are the drugs, which are immunosuppressives or immunosuppressed states. So both these cumulatively together constitute an immunocompromised state. And yeah,
Ye- Yes, sir. My next question is, what is mutation and this new mutant strain that has emerged from
Mu-
UK? What are the implications and will it make the vaccine ineffective? Sure.
Mutation is simply change. Change. Life evolved 3.8 billion years ago. From then on till today, life has been evolving. What has caused this evolution that has led to us and other diversities we see around? Mutation and its natural selection. A random mutation, why does it happen? Now, RNA viruses, any virus or any cell, including our cells, let us take this virus. Now, how does it grow?
Mm,
It enters my nose. From the nose, it
mm,
attaches to the ACE2 receptor and it enters my cell. And once it has entered my cell, what does it do? This virus has nothing in it. It's just RNA. It only knows how to make photocopies of itself, of its own RNA, of its
mm,
genome. So what it does, it captures my cell and starts making photocopies of itself, photocopies after photocopies after photocopies, and these get released through the cells and infect more cells and create more photocopies and after photocopies. Now, roughly, if you run any process, including your own photocopier machine in the office, for 1 million times, by a random error, a random chance, simple coin toss,
mm.
coin toss, a random error will occur in one for every billion such copyings. It's a random. Now, this error is what change. Now, there are four alphabets, C, T, A, G, which together create this RNA, DNA. Any change, a substitution, one A for C, or a deletion, one is dropped out, any such change, or the frame of reading has shifted, changed, these are random. This is called mutation. Mutation creates alarm. However, most 99% of the mutations are useless to the virus, useless to us. They are neutral or they are of no use to anyone. They just go by the way and they get added when we pick up the sequence, we find them. And that tells us how the virus has traveled across the world. However, once in a while, a mutation occurs which leads to either increased transmission of the virus, ease with which it transmits person to person, as it has happened in this B.1.17 strain that has arisen in Kent
Yes,
and
yes,
spread
yes.
to the UK and thereafter. It transmits easily. But remember, there are also mutations which make the virus more deadly. It kills more. Any virus that kills more will run into a dead end and destroy itself. So that will be the end. So the virus, the mutation that really thrives and survives and grows by natural selection is the mutation which creates a condition for the virus to travel fast, spread faster, and cause mild disease. I sneeze, I cough, but I keep moving and I keep transmitting, and that's what the virus wants. It wants to reproduce and keep traveling and
Move on.
occupy the
Yes.
niche, move on. So most mutations lead to nothing. Some which are of consequence lead to more transmission. Very few, if any, lead to death, increased fatalities, which lead to an end for the virus itself because it stops, it reaches its dead end, it can't transmit further. So this mutation B.1.1.7 is a mutation. The problem about this is that it has at its core a substitution N50Y. What does it mean? That in that entire genome sequence, 1 to almost 30,000, it's a 30,000 nucleotides pair sequence long virus. So from 1 to that, at the point 501, N has been replaced by Y. Technically, asparagine has been replaced by tyrosine. So it's just substitution. And what has this substitution done? It has led to a 50 to 60% increased transmissibility, its ability to capture the
Mm.
ACE2 receptors and get into the system and go move forwards. It has increased by 50 to 60% as a result of this. However, it is not as deadly as it could have been. It's not, it's mild, same as it was earlier. This has arisen together simultaneously in three different continents: South Africa, that is Africa, the Brazil, and the UK, which is very unusual. And this is it's a well-known phenomenon to science called the evolutionary convergence. Something that is of use to the virus generally will grow and propagate all over, and that's what has happened. To which have been added some other mutations which specify the Brazilian, the South African, or the UK variants. So this is there. It will not affect the virus as of today. Why? Because let us take the UK variant B.1.1.7. This has led
Mm.
to deletion of two amino acids as position 69 and 70, HV69 by 70. What does it mean? Now, the area of interest which can damage us far more is that what's called the RBD on the spike protein. RBD is what? Receptor binding domain, which is the core, the center of that spike protein, which docks to our ACE receptor cell and makes it enter our cell, the first step to infecting us. This RBD alone has more than 300 points, or what we call epitopes. These points are of interest to the antibodies which kill or neutralize this virus. Of these 300 points, just two at position 69 and 70, two amino acids have been deleted. The rest are still active. So any vaccine will be still active against the remaining sites. And if you look at the whole spike protein, there are over 3,000 such sites, base pair sites, which are still valid, open, and receptive to the attack by antibodies. So theoretically and otherwise, till now, we have not found that this mutation has the capacity to incapacitate or destroy the usefulness of vaccines till now.
Do you think India could well see some of these cases, or you are very sure that this variant will stay limited to these three places, three continents? So,
It will spread because, as I said, this mutation has made it more transmissible. The ease of transmission has gone up, augmented by a factor of by 50 to 60%. So that means that it will travel more widely across our
mm,
planet. It has already come to practically all the major continents by now. By how much magnitude it travels beyond will depend on us, our behaviors. If we still continue to wear masks, avoid crowds, especially in poorly ventilated places, maintain the social distancing, and most importantly, the mask, if we do that, we will be able to limit the spread of this virus, and that's the only tool we have until we get vaccinated. So the second strategy is vaccination, and that is why we must all go out and get ourselves vaccinated and not wait for someone else to take it before we decide to. Vaccination, social
yes.
distancing measures are the, yeah.
So one last question, sir, before we wind up today's program. How is the government planning to monitor those who receive Covaxin, sir? They're saying that this is being given under the trial mode. What do we mean by that?
Of the two approved, Covishield and Covaxin is, in addition to being restricted emergency use approval, has also got bracketed as in trial mode. Now, what does trial mode mean? That you see, if you look at our new drug and clinical trials rules 2019, you study the article
Mm,
880 and then the schedule 2. Now, schedule 2 allows for
mm,
two things: res- accelerated approval process when required under emergent conditions where no alternative is available. And the second is expeditious review process. Don't take too long. Look at the basic interim data and review and give a judgment. And 80, Article 80, Section 80 of this 2019 rules says that if an emergency situation develops where no alternative is available and it is affecting the health of the nation, then if there exists a country outside, in this case UK, where phase 3 efficacy trials have been completed, and the health regulator, the licensing regulator, in this case DCGI and CITSCO, if they find on evaluating that data of phase 3 from UK to be valid, relevant to us, to our genetic and ethnic makeup, then in view of saving lives, particularly with the new mutant coming up and threatening us at our doors, they can skip the phase 3 trials to be conducted immediately in our country and go for direct approval based on the validity of data from phase 3 trials generated in a foreign country of their of interest. Let's say this case the UK. So if that data is valid and it meets our ethnic genetic profiles, then we say in emergency use we approve. Bypassing the phase 3, they will go on and will take time, and we begin using it. That is the meaning of restricted use. However, this Covaxin, therefore, is still running the phase 3 trials. It has done completed phase 1 and phase 2 with over 700 odd, with good results, safety and efficacy. The phase 3 trials are on. The data is yet to be out. So, in view of that, it has been allowed on clinical trial mode, which means that clinical trial rules will apply. Consent, before I go to take this vaccine, I will be given an information fact sheet. Listen, these are the indications, these are the contraindications, this is all what can happen to you. Do you agree? I say, yes, I agree, I have understood. So after I've understood the information, the facts of the vaccine, I sign a consent form. I authorize them to use it on me. And in that consent form is also written that should any adverse serious effect occur as a direct cause of this vaccine, which is to be determined by the Central Ethics Committee at ICMR, then I will be entitled to a compensation by the sponsor, which is the Bharat Biotech. So the compensatory clause is there, provided causality is established between a very serious adverse effect. Otherwise, all minor and moderate effects, serious adverse effects, will be treated for free in an approved public hospital. That will be my right once I participate in this trial. The third component is the surveillance. After I have taken the vaccine, I'm observed for 30 minutes, which is when most of the problems occur, and there's a whole system to manage it. Beyond that, when I leave for the next 7 days, I'm on active surveillance, which means every day
Mm.
I have to register my health state to the authorized
Mm.
immunization officer at the vaccine site where I got the vaccine. On day 7, I submit a fully filled questionnaire format back to them for the fact that stating the fact that I'm well, or if there's something wrong, what is it? Day 7 to day 27, if there's any problem with me, I report it back to them using COVID app. So I'm under surveillance. Day 28, I report back for the second dose, which is a must absolute. And I fill up another questionnaire stating my health condition over the last preceding 28 days. I take the second shot on day
Mm.
28. And for the subsequent 35, again, they call me up and keep checking my health. And for 3 months post vaccine, first dose, 3 months, I'm under this surveillance system to monitor my health in terms of any adverse effects. So that is those are the three components of the clinical trial mode as they apply to Covaxin until their hard phase 3 data comes in. Yeah.
So happy, sir, for this very, very insightful and interesting conversation. I learned a lot for starters. Thank you so much for joining us, sir, today. For India, friends, this is a very unique opportunity from virus to vaccine, testing and tracing. And we just hope the vaccination program continues as envisaged by the GOI. On this note, this is a wrap on the edition COVID-19 Fact File. Namaskar.